Well, we are sorry for the inconvenience. We have to stop the lecture in between because of power failure. Now we'll resume our lecture. Meanwhile, we had one-to-one interaction with our speaker, Dr. Howley. I now call Dr. Howley again to resume the lecture. So please.
Thank you for waiting. And also thanks to the electricity grid. I have time to make new friends from the audience. Now let's continue to our lecture about the xylose fermenting engineering. We have three. We have totally three metabolic pathways for xylose conversion in microorganisms. Two of them are used for the engineering of yeast. One is through the oxidative pathway. Oxidative pathway is commonly used by fungus. And another way is use XI pathway, xylose isomerase pathway. It is commonly used by bacteria. And both pathways produce xylose, which is converted to D-xylose 5P by endogenous. We call it XK. And in particular, for the fungal pathway, there is a byproduct intermediate. It is xylitol. Xylitol is a major byproduct for the first pathway, the XR and XDH. It is not difficult to do the engineering strategy following the oxidoreductive pathway. Many years ago, there is an example of overexpressing of xylose reductase and the xylitol dehydrogenase genes from beta transferred into the yeast, the Saccharomyces yeast. The resulting strands grows on xylose. It can use xylose as a sole carbon source. However, the growth rate is very slow. It grows very slow. The big problem is that in this pathway, the major two enzymes they require. One requires the XR requires the NADPH, and another requires NAD plus. This problem is they form from oxidative to reductive, different forms. Sometimes they are not in balance. Abundant for this one, but maybe insufficient for this one. So it causes overproduction of xylitol, and it decreases the final total amount of ethanol production. There are a lot of new strategies to solve this problem. One is to add an external electron adapter, acceptor. The electron acceptor like this, it can be implemented into the two balls initially. Another way is to connecting another reduction. It is HMF reduction pathway. Connecting them together because in this pathway it releases the electron acceptors like this. And let's see some examples. This also they modified this pathway is ammonia simulation pathway in the recombinant Saccharomyces by deleting this gene. This gene is NADPH dependent gene. It's a dehydrogenase. It catalyzes oxidative reduction by overexpression another gene. Okay. So this way, it provides a better electron acceptor balance for the engineered yeast, and it helps the increase of final ethanol from fermentation. Another pathway is to install a functional catalase pathway. This pathway depends on the expression of two important enzymes in combination with native enzymes. The last method is to overexpression of genes including MAH and the MADPH dependent HMF reductiveness. And nowadays, I saw many authors they would like to use this method because this method simultaneously removes the level of HMF and other substances accumulated from the last treatment. Through the bacterial pathway, it is simpler and depends on the expression of cellulose isomerase. However, only this species is an anaerobic fungus. Only this one. Some other work, they finally get an engineered strain that cannot grow on xylose. Even that, the engineered strain expressed the xylose isomerase cannot grow healthily on xylose. So nowadays there are a lot of research work. They are trying to do the improvement. The most efficient way to prolong the cultivation and do the evolution in the lab. It may improve. It may improve not only the xylose function, but